This video is on Unit 5.3, Bacterial and Viral Genomics. So the first thing I'm going to do is take you on a tour of 3 different types of viruses and show you kind of how they infect cells and make copies of themselves. So the first type of virus we're going to look at is a DNA virus. So the virus itself is up here at the top. You can see it has a kind of set of purplish circles around it and inside the blue squiggly stuff. So the purplish stuff around it is called its capsid. It's kind of like its cell membrane, except the capsid is made out of proteins. The blue stuff inside it is its DNA, since that's the DNA virus. And that's all there is to the virus. As you can see, this virus is very simple. In fact, many scientists argue back and forth of whether a virus is alive or not. Most scientists would say it's not because it needs a host cell to replicate, and that's what we're going to talk about next. This whole orangish brown thing is the host. So that's the cell that the virus is going to infect. Once the virus gets into the cell, it separates into its different parts. So here are the capsid proteins, and here is the DNA that came into the cell. The DNA is really the important part here. What happens is the virus will use the copying machinery of the cell, so like the polymerases and things like that, to make its own DNA in this case. So here is the viral DNA, and to make some mRNA. So we'll label those. So here is the mRNA from the virus, and here is copies of the DNA. The DNA is going to be packaged into new little virus particles in just a moment, but the mRNA is used to make more capsid proteins. That's what these little purple things are. Okay, and then those capsid proteins and the DNA molecules come together. They join into the new virus cell or new virus particles, and they are released from the cell. So here's the new viruses. So basically the virus has hijacked the cell so that it can use its machinery to make its own DNA and to make its own proteins. The next type of virus that I'm going to talk about is an RNA virus. And here's the virus right here. You can see it looks a little bit different than the DNA virus. It still has the capsid. Okay, so it still has that purple ring of proteins there. Inside it, instead of the double-stranded blue DNA, is the red RNA. And then the extra part it has is this envelope. So this kind of brownish, yellowish ring around it with all the little purple push pins in it. That's the envelope. The push pins are the glycoproteins. So all these little purple dots around the outside of that are the glycoproteins. Glycoproteins are hopefully what they sound like. It should look familiar to you now. Glyco refers to a sugar, and protein is obviously a protein. So it's a sugar and a protein joined together. And those glycoproteins are basically little signal molecules that allow the RNA virus to locate and stick to the appropriate host cell. So again, here's the host, and those glycoproteins are going to dock onto the host cell's membrane. So they're going to attach to the membrane. They're going to leave that envelope outside of the cell, and the only thing that enters the cell is the capsid and the RNA. Now let's follow what happens to the RNA. So here's the RNA. It is actually a type of mRNA. It gets copied within the cell, and different things can happen to that mRNA. It can be copied again to be put into new virus particles, or it can be used to make different types of proteins. Here's the capsid proteins, and this is an endoplasmic reticulum, an ER, and that's where proteins go to be hooked up with sugars to make those glycoproteins. So in the endoplasmic reticulum, the glycoproteins are made. They are formed into little vesicles, which are released to the outside of the cell. So when the capsid leaves the cell, it actually takes with it a part of the cell membrane, that's the envelope, and then all those glycoproteins that were made in the endoplasmic reticulum. So now our final virus has its RNA, it has its capsid, has the envelope, and has the glycoproteins. The final type of virus I'm going to talk about is called a retrovirus, and in this case retro refers to backwards. And the reason it's considered a backwards virus is because instead of turning DNA into RNA, which typically happens in living organisms, it's going to turn RNA into DNA. So let's follow that along. Again, our virus has a capsid. This time the virus is kind of shaped like a weird triangle, so the capsid is the purple barrier around that triangle. This is a little bit smaller picture, but I think you can see it there. So it has a capsid. It also has an envelope. As you can see, it's kind of that brownish layer around the capsid. And it has glycoproteins, the little, again, push pins on the outside of the envelope. Inside the virus is the red RNA. And you might not be able to see that, but there's also little green dots, which you can see better down here, that are inside the virus. That is an enzyme called reverse transcriptase. That reverse transcriptase is able to make the RNA into DNA, which we'll talk about in a minute. This whole cell again is our host cell, is our animal cell. Okay, so the virus gets into the cell again. It disassembles into its little purple capsid proteins, into the RNA, which is the red strands, and then into the green reverse transcriptase. And then you can see right here, the reverse transcriptase attaches to the red RNA and turns it into blue DNA. Then the DNA makes a copy of itself, so it's double-stranded, and that double-stranded DNA goes into the cell's own DNA. So let's step back a little bit and put some words on this. So this green guy here, again, remember, is that reverse transcriptase, the enzyme that can make DNA. So this molecule that's made next is the RNA-DNA hybrid. So that's what we're talking about there. Then we've got double-stranded DNA. Once it gets into the host cell's DNA, which is this kind of brownish DNA here, which is in the nucleus, then this whole thing here is called a provirus. Oh, back up, not that thing. Okay, the provirus, I circled the wrong one, is this thing right here. So the DNA that's embedded in the host cell's DNA gets transcribed. So you recognize that red RNA again. That is our provirus. So that's basically viral information ready to go. And that viral information can do two things. First of all, it can just go into the virus, get packaged up, ready for the next generation of viruses. Secondly, it can be translated to make proteins for the capsid, or proteins for the glycoproteins, which again are made in the endoplasmic reticulum, exit the cell in a vesicle, put the glycoproteins on the outside of the cell, so that when that capsid leaves the cell, it can be enclosed in a nuclear, or in a membrane again, in an envelope, and then also with those glycoproteins on there. So then our final virus is all made again. So all of these viruses, these different types, as you can see, have things in common. They all have a capsid. They all have some sort of genetic material. They all need a host cell to make copies of themselves, but they are all slightly different from each other as well in how they use that host cell to make those copies. Lastly, I just wanted to show you what the variety of virus types are that are out there. So this is a table from your book, but I'm just going to sum it up on the side here as well in case you have a hard time reading it. First, we could have double-stranded, DS for double-stranded, DNA. That was the first diagram I showed you. So double-stranded DNA viruses. So they are viruses that carry that double-stranded blue DNA molecule. There are also, however, viruses that have single-stranded DNA in them. I did not go over how those work, but they do exist. The third one is double-stranded RNA. Again, I didn't go over that category, but it's another one that's out there. Fourth category, single-stranded RNA that acts as messenger RNA. So in other words, it gets into the cell and the cell can translate it to make their proteins. That is the one example that I went over for the RNA. 5, we could also have single-stranded RNA that is used to make more RNA. So put that in parentheses, makes messenger RNA. Okay, and then finally, the last one we talked about, the retroviruses, single-stranded RNA again, but this time it's used to make DNA. So those are the 6 types. Again, the ones I gave you an example of are 6, 4, and 1. In your Build a Virus project, you'll learn about some of the other types as well. Don't forget to answer the questions that go with this video when you're all done.